This is the fourth video of the Cell Death Pathways lecture. In this video, we will talk about the last type of neuronal death called autophagic cell death. So, autophagy is actually a natural and regulated mechanism of cells that removes unnecessary or dysfunctional components. And it allows the orderly degradation of recycling of cellular components. Autophagy refers to a collection of tightly regulated catabolic processes, all of which deliver cytoplasmic components to the lysosome for degradation. Autophagy is indeed a cytoprotective mechanism that enables the cells to survive in favorable conditions by reducing cellular stress. And here you can see a diagram of what kind of processes autophagy can inhibit, such as energy storage, ROS production. It inhibits genomic instability. It deals with accumulation of misfolded proteins by degradation. It also removes organelle damage, etc. So, autophagy can be actually divided into three major forms, which are commonly described as macroautophagy, which involves the sequestration of the cargo, which are large cytoplasmic components and proteins, away into a double membrane autophagosome. There is also microautophagy here, which consists in invaginations or protrusions of the lysosomal membrane here, and are used to capture the cargo. And then there is a very specialized type of autophagy called chaperone-mediated autophagy, which refers to the chaperone-dependent selection of soluble cytosolic proteins that are then targeted to lysosomes and directly translocated across the lysosome membrane through specific transporters for degradation. There are some other forms of specialized autophagy that include selective clearance of specific organelles or components, such as damaged mitochondria, and that specialized autophagy process is called mitophagy. There's also a removal of cytoplasmic bacterial pathogens, which is called xenophagy. And even there is a specialized way of autophagy to deal with protein aggregates called agrophagy. Any form of autophagy requires a very complex and orchestrated group of proteins that ensures that every step of the autophagy process is properly made. All of these proteins have a lot of different functions, and we will see some of those in this diagram. So, for instance, in macroautophagy, large cytoplasmic constituents and proteins that are damaged or no longer needed are targeted and isolated from the rest of the cell within a double membrane vesicle known as an autophagosome. The initiation of the macroautophagy pathway here begins with the formation of the phagophore assembly site, or PAS. This is a process that is mediated by different kinases, such as the UNC50-like kinase, or also known the ULK complex, and autophagy-related proteins, or ATGs. Further nucleation, phagophore membrane elongation, and autophagosome completion requires two ubiquitin-like conjugation pathways. The first produces the ATG5 to ATG12 conjugate here, which forms a multimeric complex with another ATG protein, in this case 16. And there is a second event that results in the conjugation of the phosphatidyl ethanolamine, or PE, with the LC3. the microtubule associated protein 1 light chain 3, which is actually a central protein in the autophagy pathway, functioning in autophagy, substrate selection, and autophagosome biogenesis. This LC3-PE conjugated is required for the expansion of the autophagic membranes and their ability to recognize autophagic cargoes by interacting with autophagic cargo receptors and the fusion of autophagosomes with lysosomes in this last step. The resulting autophagosome fuses with the endocytic and lysosomal compartments, ultimately leading to formation of the autolysosome, this case, which actually provides a specific and a special process of waste management and disposal. And eventually all the contents of the vesicle, now called the autolysosome, are degraded and recycled. In the microautophagy pathway, substrates are directly engulfed at the boundary of the lysosomal membrane. And in the chaperone-mediated autophagy, substrates with specific pentameric motifs described here are selectively recognized by the heat shock cognate 70 kilodaltons protein, or HSC70 here. And this is a chaperone that brings these cargo proteins with these motifs to the lysosomes. And this is dependent on a receptor called LAMP2A. And there is an interaction between this complex and the receptor and translocates the cargoes into the lysosome for degradation. In all these processes, the autophagic cargo is degraded via lysosomal hydrolases in the inside of the lysosome. Here you have two different articles you can check for more details about how all these processes are regulated. But briefly, regulation of autophagy, it's a very specific process. General autophagic flux is upregulated by nutrient responsive signals through receptors that respond to different nutrient stimulus like insulin or growth factors or amino acids or even glucose. And most notably, and this is just a summary of the several regulatory pathways that can occur, I just put here like a general idea of how global autophagy is regulated. So in this kind of like general regulation, autophagy is regulated by regulation of the ULK1 complex, by an inhibitory phosphorylation of ULK1 and 2 by the mTOR serine threonine kinase. So basically, under normal conditions, mTOR is inhibiting ULK1, which is an activator of autophagy. But under starvation conditions, nutrients deprivation, or other type of stresses, mTOR is inhibited. And now ULK1 complex becomes active, and now it can phosphorylate and activate Becklin-1 complex. This is a system. So Becklin is responsible for controlling and modulating the formation of the phagophore and the other parts of the autophagy. But Becklin is also a protein that interacts with BCL2 family members as well as the phosphoinositol 3 kinase, playing also a critical role in the regulation of not only autophagy but also cell death. So this is how whenever this pathway is affected or impaired, autophagy can become a pathogenic process of promoting cell death. One outcome of upregulation of general autophagic flux is bulk and non-selective degradation of cytoplasmic material and generation of metabolites to sustain metabolism and bridge nutrient deficits. So, in the absence of nutrients, this process is upregulated and exacerbated to promote autophagy and promote degradation of components to release and recycle nutrients that are needed for the cell. However, under pathogenic conditions, alterations in any of these steps of the autophagy process may lead into overaccumulation of damaged components or proteins that are detrimental for the cell. So, the macroautophagy pathways can be classified into different steps, as we seen in the previous slide. These steps can be classified into induction of the phagophore, cargo recognition, sequestration of the cargo, fusion with the autophagosome, and then clearance by the autolysosome. And each of these steps can be affected in different neurodegenerative diseases, and this is how it connects with cell death, or its relationship to cell death. For instance, in Alzheimer's, there is an exacerbation of the induction of phagosomes, but there are no really continuing in the nucleation and formation, and then clearance is significantly impaired. On the other side, for instance, in Huntington's and Parkinson's disease, cargo recognition is significantly affected, and so is the sequestration of cargo in HD. And this leads to an overaccumulation of misfolded and aggregated proteins inside the cells, and this is also causing alterations in other pathways that are related to autophagy via BCL2 and mitochondrial dysfunction, and this is how it relates to apoptotic induced or autophagy mediated cell death. So here in this picture you can see an example of like multiple vesicles of autophagic vesicles that are formed in brains of Huntington's and Alzheimer's disease mouse models. So these are ultrastructural analysis of dystrophic neurites, and you can see with the red arrows it shows autophagolysosomes with double membranes observed in the electron microscopy, and these are consistent with autolysosomes in which proteolysis of substrates including inner membrane it's kind of a stall, and there is an overaccumulation of these autophagic vesicles due to alterations in each of these steps of the process. So a perfect example of how important it is to know the molecular basis of these cellular processes, such as autophagy, and how you can use this information to design effective therapies to prevent or ameliorate neurodegeneration, was recently demonstrated in this paper published in Nature in late October in 2019, in which the authors used the properties of the LC3 protein, this protein that was important for autophagosome formation and cargo recognition, to develop a strategy to specifically target mutant huntingtin protein into autophagosomes, increasing mutant huntingtin clearance and improving Huntington's disease progression in mouse models of HD. So what these authors did is use their knowledge about autophagy and the properties of this LC3 protein, and they screened for possible compounds or linkers that could specifically link mutant huntingtin to this LC3. Now, because the LC3 it's bound to the autophagosome membrane and includes any substrate into the autophagosome for degradation, they were able to link LC3 with specific mutant huntingtin and promote its degradation through the microautophagy pathway. So this is again is an example of the knowledge that we gain into the specific basis of the molecular mechanisms that regulate autophagy to promote specific therapies for neurodegeneration intervention. And with that, I will leave you with a short quiz to revise some of the most important concepts that we've seen in this video. And we will see the answers for this in class. And then the last in-class problem consists on grouping different concepts to its corresponding cell death pathways, and we will see how many of these specific concepts or key molecules can be shared between different cell death pathways or which ones are unique.